So I started in San Francisco, where our lab, back in 1996, first reported on cochlear gene transfer in the cochlea. We used the guinea pig as a model. We'll talk about that. And so I had two options. I thought I could really give you the state-of-the-art molecular biology talk about gene therapy, and tell you about genes and microRNAs and all that kind of stuff. And I realized that's like giving you Ambien. You'll wonder, I just had a huge lunch. I'm going to take a little nap, I'll get to where the endoscopic ear surgery stuff comes up, because that's what you really want to know now, right? That's the current thing. How do I bring endoscopes into my otologic practice? The other approach is, and I think Doug was very smart to include this topic on the agenda, because I think whether we end up doing cochlear gene therapy, some of the things that we're going to think about, I'm going to talk about the approach to how to think about inner ear therapy. Some of those things you may be doing in your office in just a matter of years. So right now I have a nice 5-year grant from the NIH, $2.2 million, where we're trying to develop tools for delivering anything you want to the inner ear in a very controlled fashion. And the goal really is of these tools is that this will be something that'll be office-based. I think initially it'll probably be in the operating room where you'll go in and expose the round window membrane, make micro perforations, and 10 micrometers in size or something, deliver what you want to, but eventual goal is, of course, to do it in the office. From San Francisco, anyway, I went to New York, where Sean is currently, until he moves back to Seattle. So there's a nice little, one of those child structures that kind of goes around and around and around. We kind of move around and experience different parts of the country. And now I'm at Columbia. Where one of my other hats I wear is I run the perioperative services at Columbia. I run all the operating rooms, which is really fascinating in itself. If you've ever been in the hospital side of administration, very different from the academic side. Gives you a new definition to IT. I always used to think about my email, working or not working as IT. Well, that's not what IT is all about. And I've learned that, whether it's software for financial packages, for human resources, for perioperative service, and so on. So anyway, let's go on to the talk itself. So I'm going to give you sort of a way to think about gene therapy, about delivery of material to the inner ear. What are some of the things that we should be thinking about? And I'll kind of leave you where the field is right now. So why gene therapy for hearing loss? Well, we know that it's really a very common disorder, right? 10% of Americans have some sort of hearing impairment. For some, it starts very early. 1 to 4 newborns have some sort of hearing impairment. 1 to 2 have very profound, severe to profound hearing loss. And if you look at worldwide, there's about 300 million people that have hearing impairment, right? Huge impact on quality of life, huge impact on the bottom line, the bottom dollar thing, the economy and stuff like that. And yet, we don't really have an effective treatment for hearing loss. Yeah, we give them hearing aids, and occasionally surgery will help with things like otosclerosis, but we don't have a lot of things in our armamentarium that will help hearing impairment, right? So that's the reason to think about potentially something beyond what we do today. And that's the impetus to explore gene therapy, where you deliver a gene, but you can really deliver anything you want to. Whether you deliver a drug, gene, RNA, it really doesn't matter. Something that you can deliver right into the inner ear that will lead to either preserving a hearing loss that's occurring over time or recovers a hearing loss, but something that is positive, And so a few years ago, when I would put up this slide, my big point would be that this is a list of the different gene therapy trials that are going on in humans. Cystic fibrosis, clotting disorders, ischemic disease, and so on. And what wasn't on the list, even though 10% of us have hearing impairment, was hearing loss. So the most remarkable thing, I think, today is that, in fact, gene therapy trial for hearing loss is occurring. How many of you are aware that there's actually a human gene therapy trial going on right now in the US? Just raise your hand. That's pretty impressive, right? Because there's Columbia is one center. It's under Hendrix Decker from Kansas and at Hopkins. And the gene they're delivering is for severe to profound hearing loss, and we'll talk about it briefly in this presentation. It's called ATO1. It leads to hair cell regeneration. So the early results aren't very positive, but the point is that we're in the process of doing it. It's a really good safety study, but it opens up the whole landscape as you have out there, just beautiful landscape to potential for gene therapy in the cochlea. So it's a relevant topic for you because it will be part of your future. Certainly we wouldn't have predicted it 10 years ago that'd be part of our current future, and we're already here. So when we think about gene therapy, we have a variety of different ideas about exactly what that means. You know, some people think about growing new organs or something, like this big mouse, big ear on the back of a mouse. Other people think it'll make something bigger. Certainly, I wouldn't have picked ears to make it bigger. I'm more like Donald Trump, you know, like keep my hands small, but the other body part's big. If you've been following the debate, I'm glad you're current. Thanks, excellent. Or people think that gene therapy might make something. Is that Tracy Erickson in the back? Hey, Tracy. One of our residents from San Francisco many years ago. That you're going to take something and make it better. And certainly, that's not exactly what we're thinking about when we're thinking about gene therapy in the ear. In fact, what we're thinking about is that we're going to we have a state here and hearing on one side and hearing loss on the other, and we want to put a stop to that. So some of the things where we might do it are things like acquired hearing losses with aging or noise-induced hearing loss, ototoxic medications or infections, or even genetic forms of hearing impairment, where a single gene, we might correct a single gene and repair it, right? And what we're really looking forward to is a future where what already happens naturally in the chick. That is, if you're able to, in the chick, if you just give them ototoxic medication, you lose all hair cells, but over time they grow back on their own by 28 days. Unfortunately, in the mammalian cochlea, this doesn't happen. But we're trying to recapitulate that in the humans so that someday, it doesn't matter what the toxic factor is, whether it's age, noise, or ototoxic medication, that you're able to go from top to bottom and restore the sense of hearing. So there's two ways to think about how you might intervene with gene therapy. So on the left, what you have is a normal situation. You're about to be, you've been exposed to an ototoxic situation, and then gene therapy comes in and rescues it. So it's after the fact. But you can envision the other, which is, let's say you're about to go to a military service, exposed to loud noises or something, so you get a gene therapy delivered to your ear beforehand, like BDNF or a growth factor. You have that noise exposure, and you maintain your hearing. So you can either do it after the fact or before the fact. So a variety of different ways to think about when you might, in fact, deliver it. So what are the issues about inner ear gene therapy? So there's 4. And these are the questions you have to answer in terms of the issues in regards to cochlear gene therapy. One is, how are you going to get it there? All right? Second question is choice of vector. What are you going to use to deliver the whatever gene or whatever you want to deliver into the cochlea? Number 3, how about safety issues? Are there concerns about that stuff leaking from the ear or going somewhere else and causing problems? Because if you chose the herpes virus, next thing you know, you got herpes all over, you know? It's a safety issue. 4th is a choice of promoter. That's not so obvious, but often what you're going to find is that gene therapy leads to expression of that gene that you put in everywhere. But one of the ways to regulate where that gene is expressed is by something called a promoter. We're going to briefly just touch on that. So let's take a look at the route of delivery. There's three broad categories. One is systemic delivery, systemic injection. The problem with systemic injection is very little actually gets into the cochlea. So you have all the systemic toxicity, but very little actually going in. The middle categories are the intratympanic delivery, where you're relying on the capability of diffusing across the round window membrane. We're going to discuss those. But ultimately, the barrier is the round window membrane. So I'm going to spend a little bit of time about how do we overcome the barriers of the round window membrane which is the last category, which is direct intracochlear delivery. So people, for example, middle ear space delivery, people have used something called a Silverstein-Oda wick, by and large, not very helpful. You put a tube in, put a wick in, it goes right next to the round window membrane, you drip some drugs, medication, something. It doesn't really work very well, and not too many. Anybody used a Silverstein-Oda wick, by the way, in the audience? Not very successful. Transtympanic injections, we all do it, right? How many of you believe intratympanic steroids work? How many believe you don't know? And so part of the problem is the answer really isn't there. We don't know how much, when they don't work, is it because the steroids didn't get in? Or they got in but actually didn't work? So we actually, remember that oral steroids didn't even make it onto the Academy's list of recommended intervention for sudden hearing loss, while intratympanic steroids did as a salvage. But the data is pretty weak, and it's large part because we don't have ways to reliably deliver and measure what's inside the inner ear when you put it in the middle ear space. There were all sorts of these direct pumps that were placed against the round window membrane, and they're off the market now. They were FDA approved for a while. And so the next mode is, of course, direct delivery to the inner ear. So you can bypass the round window membrane. The last thing, of course, before I get to that is you could place a gel foam right adjacent to the round window membrane. Have you guys heard of Otology, the recent trials where you have a liquid with steroids in it, you inject it in the middle ear space, and at body temperature, the liquid becomes solid. And the purpose of that is to deliver those steroids or gentamicin, whatever you might want to deliver, over a prolonged period of time. Gel foam does something very similar. You put a gel foam against the round window membrane, it's going to constantly soak up material and hopefully allow diffusion to occur over a prolonged period of time. They all work, but they don't work great. So let's go to direct injection or a cochleostomy or a microinjection. And that's kind of where we started our work back in the early 1990s when we did our first gene therapy experiments. And so what we did at that time is made a little cochleostomy in a guinea pig cochlea, about that size. Used the LZ osmotic mini pump attached to it because we wanted to be able to deliver a lot of gene therapy vector in there because we weren't sure if it was going to work or not, but we didn't want the dosage of the vector to be a problem, right? So we wanted to this infused at about a microliter an hour or something. So nice slow rate, physiologic. And what we found was that the mini pump through a cochleostomy did a very nice job of diffusing material to the cochlea. And there was relative cochlear morphology preservation, with the exception that right where you made your cochleostomy, there was a little bit of fibrosis. So when you cause a little bit of damage or cochleostomy during cochlear implants, you do create localized injury, right? What happens to the hearing when you make that hole in the ear and put a pump in it and let it infuse over time? Now remember, this is early 1990s, before the hybrid cochlear implants were around. We were deathly afraid of doing anything to the inner ear, right? Because we thought anytime we would do anything to the inner ear, it would be death. So what was remarkable is that when we tested the hearing of these mice, guinea pigs, with a click, broadband noise, so here's pre-op hearing around 60 decibels, so it's a relative decibel threshold. Post-operatively, the most remarkable thing at 3 days and 7 days is they weren't deaf. It was elevated, we lost hearing, but they weren't deaf. So the next thing we did was we measured frequency-specific ABR thresholds. So what this shows is that this is the pre-op level from low frequency to high frequency, post-op at 3 and 7 days. And what you see is that the hearing loss is primarily in the high frequency where the cochleostomy was made, whereas the low frequencies are relatively preserved. So that if you can minimize trauma when you're delivering material to the inner ear tolerates it pretty well. And in fact, if you think about it, that opens up the whole era of inner ear interventions, whether it's diagnostic purposes, whether it's therapeutic purposes. What it tells you is if you minimize trauma, there's a lot you can do to the inner ear and not lose hearing. And in fact, when we did the same study using microinjection, injecting about 10 microliters over a course of an hour, there was barely any change in hearing whatsoever. Okay? So minimize trauma, don't lose hearing. A little bit more trauma, you lose hearing. So you can control a great deal in terms of what that intervention leads to. And what's really kind of cool is now we know that we can do the same thing with a hybrid cochlear implant, right? The shorter implant where you're supposed to, where in the older patients, the high frequencies are gone, but the low frequencies are still there. So you put a shorter electrode, which stimulates only the high frequencies. And what's remarkable about that even though you put an electrode into the cochlea, those low frequency hearing is still preserved. It's only a matter of time that we're going to aspirate material from the inner ear for diagnostic purposes so that you can practice precision medicine or personalized medicine. And we're designing the tools for you to do that with. We did gel foams as well, and that was the best, but it didn't get a lot of stuff in there. So I'm going to tell you a little bit about stuff that we're doing with microneedles. And so right now in 2016, we lack a reliable and predictable way to deliver material to the inner ear without causing anatomic or physiologic changes. So we had an idea of trying to create microneedles because we know the round window membrane, if you make a little nick in it, it heals, right? If you make a nick in the round window membrane, it doesn't cause deafness. If you make a big nick, don't close it off. You could have a perilymphatic fistula, you could lose hearing and all sorts of stuff. But you can make little holes in the round window membrane, and they heal over time, depending on how large it is, in a few hours to a few days. So what would happen if you created this 10 by 10 array of microneedles about 20 micrometers in size, all right? So we're envisioning something like this. We use the microneedle, not going to look exactly like that, and that creates little holes like this. Holes that occupy less than 1% of the surface of the round window membrane. And you put whatever you want to on this perforation right here, and you let it go in. So we did some calculations, and we found that if you make holes that occupy only 1% of the surface area of the membrane, you increase the diffusion rate by 35 to 50-fold. Okay? That's mathematically. And so we did some experiments to do exactly that. And I don't believe this video works. But anyway, this is a 20 micrometer needle that we manually made a hole into the round window membrane in the guinea pig. Because yes, we could use robotics, but how many of you are going to have a robot to make a little micro perforation? So if you can do it manually, make a manual perforation that occupies less than 1% of the membrane, 0.22%. And what we showed is a 35-fold increase in the permeability of the membrane. So what we had determined before doing the experiment mathematically, in fact, was borne out in experiments in vitro. So we're doing the same stuff now in vivo. We're measuring the hearing and so on. And our early stuff shows exactly what we expected. The holes close up, hearing doesn't change, and you increase perfusion. And we're designing not only these needles for passive diffusion, but also hollow needles for active diffusion. So what's really remarkable, this was a kind of surprising thing. What's remarkable about this, that if you think that, if you can inject something in, it'll go at a faster rate than if you simply create passive diffusion. It turns out at 20 micrometer size, at 10 micrometers, there's no difference in the rate of delivery because of the resistance to flow. So the advantage of hollow needles over time is you can deliver it over a prolonged period of time. Okay? And this sort of is a cartoon that shows that here is a diffusion going on, barely a few molecules go in. That's the lower one right here. And this is what happens when you create a tiny perforation. It's much greater diffusion. And the one thing I forgot to mention in the last slide, coefficient of variance. How much variability do you see when you simply let diffusion work versus introducing a single hole? A lot of variability. Remember when the hands went up, half the people thought it might work when you put steroids, the other half didn't? That's why. you don't have a reliable, predictable way of delivering the stuff you're putting in the middle ear into the inner ear. That's why all the skepticism and putting the hands up about knowing or not knowing. So the next thing is, well, we made one hole and increased it. Does it make a clinical difference, right? So let's say we put gentamicin on an intact round window membrane, and I forgot the exact doses we're trying to target inner ear, but let's say we target a reasonable dose. It takes 7 days to get that in reliably based on diffusion alone. You make one hole like we just did, that cuts it down to 4.5 hours. Still a long time. You sit there in the operating room for 4.5 hours, that's not going to happen, right? You make 3 holes, and it turns out mathematically this is a linear thing. It goes down to 1.5 hours. And if you make like 9 holes, it's a half hour. So you can envision either in the office or in the operating room, you go in, use this tool to make microperforation, and we got cute little things on there too, that the moment you go across the membrane, it goes beep, because it can detect a change in resistance. So not only do you have visual inspection, but you also have an electrical way to know when you're in. You deliver whatever you want to deliver, because these holes are big enough for gentamicin, steroids, stem cells, RNA, DNA, it really doesn't matter what you want to put in. You're going to be doing this, especially those residents back there. You're going to be doing this in your practice, because you're going to have great imaging with fiber optic scopes, and you're going to do this in the office and deliver medicines when you have severe to profound hearing loss or Meniere's disease. So I think the microneedles is the future. This is what our grant's about. We're about to put our first product to create very precise holes for cochlear implantation. Because now that we're doing hybrids at 0.4, all those electrodes are really tiny. And we have a spinal needle making a huge slit in the round of membrane. That's not precise. So we're creating a nice serrated needle that we're doing human trials on right now. So in terms of route of delivery, there's a variety of techniques. There's advantages and disadvantages. But we prefer something that's non-invasive. But if it's going to be invasive, it's going to have to be safe and reliable. And I think microneedles will offer us that. So the next question is vector. What are you going to use to deliver the medication in? So by and large, you're probably familiar with a variety of viruses out there. The earliest ones, the most commonly used ones, were retroviruses. They were really used for cancer cells that were dividing because they go and infect dividing cells, but hair cells don't divide. So retroviruses aren't great for us, even though lentivirus is a retrovirus, and we'll talk about that. Adenoviruses is a good vector. The problem with adenoviruses, first of all, it doesn't integrate into the cell, and therefore the expression is temporary. And more importantly, it causes an immune response. So do you really want to put a vector in that causes an immune response where your hearing loss is worse afterwards than before? That's a concern, right? Herpes? Well, I don't want to get injected by herpes. I don't know about you guys. It certainly would raise some eyebrows. Now, I'm going to talk about adeno-associated virus, which is what we chose, and the current human gene therapy trial is also with AAV. But the ideal thing is going to be a combination, sort of a hybrid vector. It'll be based on some kind of a liposome, that is lipid membrane, combined with the infectivity of viruses. Okay? That's what we're going to want. Because we don't want the risks of what comes with the viruses. We want the safety of the liposomes with the infectivity of the viruses. So we worked with AAV. Turns out AAV is able to integrate into non-dividing cells, not known to cause major disease. The biggest thing is you can't put a very big gene in there. You can only put about 4.5 kilobases. The average gene is about 2,000 bases, but there are big, huge, like myosin is huge. When we injected beta-galactosidase in a guinea pig, we showed that all the brown stuff is where the gene expresses everywhere. We showed that it was there for 24 weeks, the longest studies ever done, in the cochlea, in the vestibular organ, in the macula. And then we did different things. Liposome worked the same, AAVs right there. When we put the lentivirus in, the expression was only in the perilymphatic space. So you might ask, well, is it because lentiviruses can infect the other cells? The other possibility is it never got out of the space, right? There's barriers for stuff to leak from the perilymph to the endolymph. And it turns out that's the problem. Lentivirus is huge compared to adeno-associated virus. And it's restricted to space because we showed if you put it on separately on auditory neurons and hair cells, not hair cells, but auditory neurons, it's able to infect those things. So size matters in the vector as well. And really, each of these viruses have various characteristics, but AAV is a really good one because it's safe. But other ones work as well. I can't advance it. Then the next thing we did was we put AAV with BDNF and showed that we can enhance spiral ganglion cell survival. These are all the different neurites after treatment with amikacin. So it can rescue auditory neurons. Gene therapy does work. So the next question is safety. So when we did these experiments, we injected this stuff, and we used the contralateral cochlea as a control. And for the life of me, we couldn't get the background staining done, get out. And we thought, we're clinicians trying to do science. We suck at doing science. We can't even get rid of background staining on the other side. Then we tested the hypothesis that maybe it's not background staining. Maybe the virus is getting from one side to the other, right? 3 potential ways. One is through the blood. Turns out that's not the way it gets out. The other 2 possibilities, the bone marrow space. The bone marrow space of the temporal bone is connected from one side to the other. And number 2, the cochlear aqueduct. So in guinea pigs, the cochlear aqueduct is wide open, where it's not in humans. And in fact, Yoash Raphael nicely showed he could track the blue staining from one side of the cochlea to the other to the brain. And we showed that, in fact, in bone marrow space, you can have staining from one side to the other. So there are safety issues. And we got rid of this problem entirely by microinjections. That is, once we injected and then used that infusion pump, where we had ongoing delivery, we didn't see the contralateral side. But it shows you that you can't use the contralateral side as control in most of your experiments. So you actually have to use a separate animal. And finally, a choice of promoter. So remember that everything is brown, right? I showed you the cochlea, everything is brown. What we want is that even though every cell has that foreign gene in there, you only want to see it expressed in whatever cell you're interested in, for example, the hair cell. And the way you do that is a promoter. That's what drives the expression of a gene. So if you have a promoter that only works on the hair cells, the gene's only expressed in the hair cells. If you have a promoter that only works in the auditory neurons, it only expresses in auditory neurons because you don't want the toxicity of expressing the gene in all the different things. So what might we do once we have all those tools at our hand? Let's say we've answered all those questions now. Well, we might regenerate hair cells. And how might we do that? We might either put in an agent that leads to hair cell regeneration. We might put in stem cell therapy that leads to hair cell regeneration. We might put an agent in that is preventing hair cells from dying and so on. And we can do that because we know a lot about the cell cycle of hair cells and how they're developed, what cells give rise to them. And so, for example, if you look at these different genes on this side, if you knock out the HES1 gene, for example, you get too many inner hair cells. If you knock out the HES5 gene, you get too many outer hair cells. The gene that we want to talk about is MAT1. MAT1, you knock that out, no hair cells. You give MAT1 back, you get lots of hair cells everywhere. And in fact, they did those experiments. They showed that you can, this is a knockout that's just showing all the hair cells are gone. You deliver it, and you get hair cell regeneration. And more importantly, even without giving additional direction, the neurons come to that new hair cells. So that is, you don't have to know everything about cochlea and how it works. It might be just injecting and taking care of one problem will take care of everything else. And in fact, they showed that they could preserve hearing with the hair cell regeneration in that system. And finally, the last thing I want to talk about is this nice paper from UCSF, from Larry Lustig's group, where they gave a gene for a replaced VGT3, glutamate transporter thing. Glutamate is released from inner hair cells. Glutamate knockouts lead to loss of hair cells or death.
They gave it either at day 1 or day 10, and they found that giving it at day 1 led to much higher expression of the VGLU3 as opposed to day 10, led to restoration of hearing. It lasted longer if you gave it early versus late. So, I think the future is just around the corner. Gene therapy can be safe, it can be effective, it can be specific, if you make the right choices. And the future is so very bright. I don't think this will happen. I think every era will give us new opportunities to intervene, because even though we always think we're going to replace ourselves, that's just not going to happen. So I think this is a great, bright future. Thank you very much.